Hello, people. I hope you're keeping warm. My wife has got our thermostat down, so I'll be lecturing like this today.
Why is the thermostat down?
To save money. I'm a professor, so I'm pretty rich already, but still she thinks we're not rich. So here we are, freezing our butts off.
It never hurts to be frugal, even when you're as wealthy as you are.
That's true. That's true. I guess that's why I'm so rich.
I have a sister who lives in Houston. And so she has been without electricity since 2 a.m. the night before last.
Oh my God.
And it's 40 degrees in her house.
Oh, wow. Wow, that's bad.
She's got two little kids. Yeah, no, it's a bad scene.
It probably has no fire either. I mean, we've got a fire we can get going if we lose electricity, but
yeah, they don't even really have jackets. I mean, they just don't have much in the way of like keeping warm. I think we're glad for.
Wow, that's horrible.
Two of my kids are in Houston.
Really?
And yeah, one's a firefighter, so he gets to be out in all of the muck and stuff. And yeah,
oh my gosh.
My daughter sent me videos of her sledding, and yeah, they're having a blast. They're having a blast because they don't have to go anywhere.
Does she have a sled? What is she sledding on?
I don't know. It looks like a garbage can lid.
Okay, yeah. I was going to say you wouldn't go out and buy one.
No. Yeah. My son lives actually in Conroe, which is north. No, he lives in Montgomery, which is north of Houston, and my daughter lives in like the northwest part of Houston. So yeah, okay. They're this is all kind of new to them. So. Yeah.
I have to admit, funny. Feeling a bit of schadenfreude. I do. We've been freezing our butts off for months. Of course, we do every year, so we're used to it.
Yeah. So it's all. Their dog is having a blast. She's digging in the snow and making all kinds of snow.
Yeah.
Yeah.
Okay, so I only have 3 more things I want to talk about today, and then I will review if you guys have some questions. I don't have anything specific to review about, but if you have questions, I'll stick around and answer all your questions. So, with no more ado, let us go to, where is it? There it is. Smooth muscle contraction. There's my dog showing up right on time. I always look at my notes so I don't forget anything. By the way, careful with these notes when you download them. The ink is still wet. I finished them about 10 minutes ago. Metaphorical ink, I should say. Okay, smooth muscle contraction. So smooth muscle is a lot different from cardiac and skeletal. If you remember, cardiac and skeletal were both striated muscles, and the contractile apparatus is pretty much the same in both of them. The big difference is how the calcium is released from the SR. With skeletal muscle being the direct connection between the voltage-dependent calcium channel and the ryanodine receptor, and cardiac being calcium-induced calcium release. So other than that, they're pretty much the same. Smooth muscle, very, very different. It doesn't have striations, which is why we call it smooth muscle, right? If you look at the cells here, they're not striated. They're also smaller. I guess they're about the same size as a cardiac cell. They're not long like the skeletal muscle cell. Kind of spindle shaped, okay, thin at either end and thick in the middle. And they've got these dense bodies that are connected to they're made of intermediary filaments, and they're connected to the cytoskeleton on the inside, and they help the thing to contract. So how actually is it contracting? I'm glad you asked. It is, what else did I want to say? Oh, so one thing, what's really different about this is you don't have that contractile apparatus. And so And also the myosin and actin are both regulated. In skeletal muscle, the actin was regulated, right? We had tropomyosin that was held over the myosin binding sites, and the myosin was always ready to go. Here in smooth muscle, the actin microfilaments are still blocked, so they can't, or the microfilaments are blocked, so they can't, they're inactive at rest. But the myosin is also inactive at rest. It's turned off. And the regulation of the myosin, oops, that's not it. So the regulation of myosin then is based on the folding up of the myosin. So here you can see it here. So this is myosin when it's relaxed, and when it's activated to contract, it elongates. So that big long tail is folded up during relaxation. It comes back out. And what makes that happen is phosphorylation of the myosin light chains. If you remember way back when we talked about myosin, I said there's the globular head, the hinge, and the tail, but then there's these little light chains that regulate the myosin head. Typically in skeletal and cardiac, we don't talk much about the myosin light chains. I'm not really sure, frankly, what they do. They may regulate it to give you a stronger contraction. I don't really know. But in smooth muscle, they're vital. So in smooth muscle, the phosphorylation of those myosin light chains causes the myosin to unfold, and then the tails of different myosin light chains interact with each other. Let's see if I can find that from here. So once they unfold, they interact with each other like this, right? And then they grab onto actin microfilaments, and then they can, walk along the microfilaments and cause a shortening of the cell. So in the relaxed form, they are folded up, and then in the contracting form, they spread out and they connect to each other, and then they connect to the microfilaments. The microfilaments, on the other hand, and this is something your book doesn't talk about.
Sorry, could you close the format background panel there, just so, can you see on PowerPoint?
Oh, you mean like
right on the right? I mean, we can't see most of the slide because it says oh, there we go. Well,
I see. Yeah, I see what you're talking about. Boom.
Oh, much nicer. Thank you.
How's that? Okay. Okay, so thanks for telling me, by the way. Okay, so that's how the myosin is regulated, and that's all calcium based. I'll talk about that in a second. Also, the actin is regulated. In skeletal and cardiac, we had tropomyosin across those myosin binding sites. Different protein in smooth muscle called caldesmin, and caldesmin does the same thing. It binds to the microfilament and blocks the interaction of myosin and actin by covering the myosin binding sites on the microfilament. Caldesmin will also be calcium regulated, controlled by calcium, as well as phosphorylation, but we don't need to talk about that right now. I'll show you that when we get to the figure. So basically, big difference with smooth muscle. You've got control on both the myosin and the actin. Myosin is controlled by phosphorylation of the myosin light chains. And here's a word you should know, keep in your mind, because I talk about it all the time. The word is kinase. Kinase is an enzyme that adds phosphate to a protein by using ATP. So it uses ATP to add phosphate to a protein. That's a kinase. And then the opposite reaction is a phosphatase that takes the phosphate off. And this is it's so goddamn vital in cellular biology, phosphorylation. It's so ubiquitous. Usually it's like you phosphorylate a protein, you turn it on, you dephosphorylate it, you turn it off. Or, phosphorylation might turn it off and dephosphorylation turns it on. So phosphorylation is an on-off switch for proteins. And the reason is, if you know what phosphate looks like, it's like phosphorus with 4 oxygens around it, and phosphate by itself has got, at cellular pH, 2.5 negative charges. So you stick that on a protein, now suddenly you've got this big, bulky, negatively charged thing attached to the protein, and the whole protein changes to move to a new conformation to make it more stable. So phosphorylation, dephosphorylation turns things on like that. So anyway, with our myosin light chains, they're going to be activated by a specific enzyme called myosin light chain kinase. So myosin light chain kinase, MLCK.
What did he say was the opposite of kinase? The one that dephosphorylates it?
Phosphatase. Phosphatase. And we'll talk about phosphatases in this class as well. Okay, so we've got caldesmin on the actin, and we've got dephosphorylated myosin light chains, and now we're going to contract. So let me show you this slide. This is my fancy PowerPoint animation. So up here is a signal transduction mechanism that increases calcium in the cell. I'm not worried about that for this chapter because we go over it in depth in, I think it's chapter 23. So I'm not really worried about this part, but basically a hormone hits a receptor, and the receptor then activates a G protein. G protein activates an enzyme, phospholipase C, which then breaks down PIP2 into IP3 and DAG. So we'll talk about that a couple chapters from now. Don't worry about it that much. What's most important here is we get release of calcium from the ER, from the smooth endoplasmic reticulum. Okay, so this is where we're starting in this class. Calcium increases inside the smooth muscle cell. And also, calcium could increase because of a depolarization of the membrane. The membrane has got voltage spin calcium channels in it, just like cardiac tissue does. And if the membrane depolarizes slightly, they open up and calcium can come in that way. So bottom line is the beginning of the contraction is increase in calcium. So calcium then will bind to calmodulin. Calmodulin is a calcium binding protein. It binds four calcium ions, and when it does, it changes conformation, and it will then bind to other proteins and activate them. One of the things that will activate is myosin light chain kinase, and over here is our myosin all folded up, and those little yellow things are the light chains. So myosin light chain kinase, when calmodulin binds to it, will become active, and it will phosphorylate those light chains. When it does, they straighten out, and they attach to each other, and now they're ready to contract. So the control of the myosin light chains then has to do, or the myosin has to do with calcium increasing, binding to calmodulin, activating myosin light chain kinase, phosphorylating the light chains, and you're ready to go. Here on the microfilament side, we've got caldesmin blocking the myosin binding sites. So one way to remove that is to have calcium calmodulin bind to caldesmin and pull it off. Okay, that's not what I'm showing in this figure, but that also happens. Calmodulin binds to it, pulls it off. Another way is to activate an enzyme, protein kinase C, which is also activated by calcium. And protein kinase C will phosphorylate caldesmin, bringing it off, and now the actin and myosin can interact. So again, to kind of, I stole this figure from a different class I do, cardiovascular pharmacology, where I talk more about all these things. For this class, remember this: calcium increases, binds to calmodulin. Calmodulin activates this pathway, activating the myosin, and calmodulin can also activate this by binding to caldesmin and bringing it off, and now myosin and actin can interact. So calcium increases, the cell contracts. And then we also have our circa over here on the smooth endoplasmic reticulum. And so when the cell's no longer stimulated, calcium will decrease, and then all this stuff reverses. Your phosphatases will come and take the phosphates off the myosins, and they will curl back up like they were before. Okay, so that's smooth muscle contraction. A bit more, I don't know if it's more complicated. It's very different from smooth or cardiac and skeletal. Any questions? Anyone want to write a chat question?
What is the cross-bridge cycling?
So cross-bridge cycling we talked about with skeletal muscle, and here that's pretty much the same in smooth, and that's this bit where myosin binds actin, or myosin binds ATP, lets go of the actin, phosphorylates itself and changes position, and then it grabs the actin again and then pulls it and just cycles through that cycle, grabbing and pulling, just like pulling on a rope, pulling a rope towards yourself. Keep cycling. Okay? Is that good, person who asked the question?
Yeah, thank you.
Okay, good.
Follow-up question.
Mhm.
This cross-bridge cycling, where would you mostly find it? Like in the smooth muscles or in the cardio muscles? I don't know.
This is kind of a general thing. It happens whenever you've got actin-based movement. You've got myosin and actin doing cross-bridge cycling. So in the sarcomere of skeletal muscle, the sarcomere of cardiac muscle, you see it. You see it here in smooth muscle. We will see it in a minute when we're talking about cell crawling. Anytime there's an microfilament-based movement, myosin is involved, and it's doing this cross-bridge cycling. That's what's causing it. Okay? And in the same way, whenever you've got microtubule-based movement, it's either dynein or kinesin, and they're basically cycling just like myosin does. Okay?
Stupid question, but I kind of wanted to know.
No, it's not a stupid question. I probably should have made that more clear at the beginning. I wouldn't say it was a stupid question. Okay. Any other questions? Okay, so.
You said it's like the microtubule based movement with the dynein and the
Kinesin.
kinesin.
Yeah.
Cross bridge.
Right, like they don't call that cross-bridge cycling, but it's pretty much the same. If you look at what's going on in the microfilament, you've got two microtubules, and the dynein of one is grabbing this and walking along it, right? So it's kind of like cross-bridge cycling there in the microtubule. It's not called that though, but it's a similar kind of process where you've got two little legs coming out, grabbing and doing this.
Okay, yeah, thank you.
Okay, so last two things to talk about, and in your mind, keep these two things separate, okay? Because people confuse them, and that causes trouble. One is cell crawling, which a lot of cells can do, and the other is amoeboid movement, where there aren't that many cells that really do this. Amoeboid movement is used by, believe it or not, amoeba. Amoeba is the number one, but also there are some leukocytes called macrophages that move like this too. Okay, and they do that because as these pseudopods go out, they surround bacteria and eat them up in wounds and stuff. So it's really important for them. So anyway, actin-based movement. Let's watch a video here. I'm going to have to change my share. Hang on a second. Stop. Come on, baby. Come on. All right, I'll just stop. This is kind of like 70s porn music. I don't know. It always makes me uncomfortable. Okay, you see that ruffling edge out front? That is the lamellipodium. Okay, so it's going kind of fast, but let me show you what you should be focusing on here. This ruffling front edge is the lamellipodium. So that's reaching out and grabbing and pulling itself. And then back here, it's letting go of its attachments back here. So let's watch that again. Go. So it's just kind of repeating itself over and over again. That's cell crawling. Let me try another one.
Is it fully like letting go of the attachment?
I'm sorry, is it what?
Is it fully letting go of the attachment? Like, is the attachment like?
Good question. It's supposed to. Sometimes in vitro, when you're doing this on a slide, it can't let go, and so a chunk of the cell breaks off. In vivo, it would be letting go in the back edge.
Thank you.
Yeah, sometimes when they're crawling across glass, that connection is tighter than it should be, and a chunk comes off. Okay, can you guys see this new one coming up? Yes? Okay. All right, so this is neat. It's very, very short. But the neat thing about this is there's a bacterium out here, and this cell is crawling towards it. And that's kind of important because that's chemoattraction. So it's being attracted by some chemicals being released by that bacterium. Here's your big old lamellipodium out front, and it's being dragged in this direction. Let's watch that again real quick. Oh, grabs that bacterium.
What exactly is it going to do with the bacteria?
This is a white blood cell, so basically it's eating it up to get rid of it, So it's protecting against infection that way.
So is a lamellipodium just basically a white blood cell?
A lamellipodium is a foot that's put out by a cell. It could be a white blood cell. It could be a different cell. Fibroblasts will crawl this way. A lot of cells will crawl this way. Later, we'll talk about early development in the xenopus, in the frog, in a frog. And cells are crawling, they're moving around. This is what they're using to crawl, is cell crawling. But a lot of cells can do this.
Does it like have the same function of like a lysosome?
Well, once it swallows it up, yeah. Once this cell grabs that bacterium, it will phagocytose it, bring it in, and then fuse that bacterium with lysosomes and then degrade the bacterium. We'll talk about that in chapter 8, which is next.
Okay.
I just want to say that I watched these videos the other day, and I used the gear, the settings gear in the lower right-hand corner of the YouTube screen, and I watched them at like 0.25 speed.
Ooh.
Like, it's just really cool to watch them, especially that last like disco porno one.
Yeah.
Like it's really cool to watch them like slowed way down.
0.1, no, this way, 0.25?
Yeah.
I didn't even know you could do that. Thanks for telling me. Oops, let's crop. So you can see the rippling here out front as it's pulling, letting go at its trailing edge. Okay, let's look at that last one again. I shouldn't call it 70s porn because I'm probably going to get reported to Title IX for some kind of infraction for that. But such is life. Okay, let's watch this one on slow. The music sounds better on slow too. Wow, suddenly it's death metal. Yeah, you can see all this rippling out here. See, there's a chunk that came off. You know what it sounds like? 9-inch nails now. If it was a little bit faster, it would be 9-inch nails.
So how exactly does the rippling cause the like attachment to like let go?
I'm glad you asked. That's the next thing we're going to do. That's the next thing we're going to talk about. Okay, so what is the process of cell crawling? What, in the name of all that's holy, is going on here? Hello, cat. Here's the ubiquitous cat zoom. Everybody with a cat knows their cats love to zoom. Okay, so before I answer that question, let's talk about why. So the reason these things are crawling like this is pretty much always chemoattraction. So you've got some site source of a chemical. It could be a bacterium that's giving off toxins that are attracting it, or it could be some cells that are in an area of damage. So say you get a cut, right? Cells will crawl into that area and then release cytokines, growth factors, and other hormones. And other cells will sense that and crawl towards it to help repair that damage. So the idea is you've got some site source of a chemical, and because it's coming from a pin source like this, there's a gradient of it as you go away from it, right? It's highest at the source and lower as you go away. So your chemotactic cell will sense that and crawl towards the source. So what that chemical is doing is activating receptors in the membrane, and the receptors are then activating G proteins. And we talked about these G proteins in chapter 13, RAC-RO, CDC42. So the G proteins then will activate the production of all these structures that we talked about in chapter 13. Let me pull up my notes real quick. Let's share my notes. I'm losing it. I swear to God, this Zoom stuff is just getting to be too much. Okay, so it's regulated by these G proteins, RAC-RO and CDC42. If you remember from 13, RAC makes lamellipodia, RO makes stress fibers, and CDC42 forms filopodia. Now, at the time, someone asked this question, didn't have an answer, still don't. In chapter 13, it talks about CDC42 being involved in branching. And branching is important for lamellipodia, but not for filopodia. So there's a bit of a discrepancy there in your book, and honestly, I don't know the answer to that, so let's just pretend it's not there. Okay, so just ignore this and focus on this, okay?
Oh, for the test, branching would be useful for lamellipodium.
Yes, exactly. Branching happens in lamellipodia, yep. But on the test, I won't ask you which G protein causes branching. Actually, I think that is in the game. I don't know. But I'll be understanding on the exam. I wouldn't take points off for you for doing something I told you to do. I hope not anyway. All right, but anyway, when the chemical, the hormone usually activates the cell, it activates these G proteins, and that forms the 3 different structures that are necessary for it to crawl. And these G proteins are activating ARPs, which we talked about in 13. Things like profilin, formin, cofilin, thymosin beta 4, or all those proteins get activated by these G proteins. And wasps involved in the branching. And then you're going to remodel the cytoskeleton. So remodel is a word that's used a lot in cell biology now. Basically means tearing it down and rebuilding it in a different way. So these ARPs then are going to depolymerize filaments in one place, move the monomers, and then repolymerize them in a different place to make these structures. And these receptors are activated by a gradient of the chemical that's coming from a source. So these are the 2 examples I talked about. And then the cell is then activated to move towards that source. So the 4 steps then of moving, to answer the question that was asked earlier, are here. Number 1 is protrusion. So protrusion involves the production of lamellipodia. So as these microtubule or microfilaments are polymerized, they push the plasma membrane out. Okay, so the plasma membrane gets stretched out by that polymerization. And inside this lamellipodium now, remember I kept saying, oh, look at the riffling, look at that thing riffle. The riffling is before it attaches, you get treadmilling going on in there. We talked about treadmilling of microtubules. We see it here in vivo with microfilaments. You don't see it in vivo with microtubules. So here it's basically you're polymerizing at the front edge and then depolymerizing at the back. And then you force those actin monomers forward and you polymerize, and then you depolymerize at the back. So basically you're polymerizing out here, depolymerizing back here. It's treadmilling. And it's going to keep treadmilling until step 2, make a new attachment. So it'll riffle until it grabs, and then the cell can pull itself forward.
I'm sorry, did you say when it's depolymerizing, is it using the actin from the depolymerization on the forward, like on the polymerization that's going on in the front? Is that that's recycling?
Right, exactly. The monomers come off the depolymerization, and then through contraction of the stress fibers, basically you get pressure behind it. They get forced through, and then they get polymerized at the other side.
Okay, great.
Okay, so eventually though, it's going to attach. Now the way it attaches is through these proteins called integrins. Integrins are membrane spanning proteins, right? They're both inside and outside. On the inside, they'll attach to the cytoskeleton. On the outside, they attach to the extracellular matrix. So here you can see attachment to the extracellular matrix. So that's mostly fibronectin, also some collagen and stuff. On the inside, you've got other proteins like talin and vinculin. These act kind of like ankyrin in that they attach the integrin to microfilaments. And then you've got alpha-actinin cross-linking these microfilaments. This is alpha-actinin from back in chapter 13. It's just a bundling protein. Okay, so you get all these actin filaments cross-linked by that alpha-actinin. So they form like a big rope here. And then this structure here is basically all the proteins of this focal adhesion. So instead of having integrins all along the membrane, they're all focused in one spot. And we'll talk about later in, I believe it's chapter 24 about cell division. In addition to all these structural proteins that are holding this structure together, you've got signal transduction molecules here. Focal adhesion kinase is one. And when the integrins grab extracellular matrix, it'll cause one type of cell signal transduction. When they let go, it'll do something else. And that's why if cells are not connected to extracellular matrix at all, they'll die. They go into apoptosis and die. So there's a lot of really important signal transduction going on here in both directions. So if it's not connected to the cytoskeleton, it'll die. But also the inside of the cell can signal these focal adhesions to let go. And we'll see that in step 4 of cell crawling. Okay, so bottom line is after it's reached out, it makes a new adhesion, and it does those adhesions through these focal adhesions using integrins. And step 3 then is translocation. It pulls itself forward, and that's stress fibers. So you've got those stress fibers crossing the cell, and myosin interacting with those stress fibers and pulling them towards each other. That kind of shrinks the cell in the direction it needs to go. And then at the tail end, it lets go, or at least it should. Like someone noted before, sometimes a chunk gets lost. I think that's more in vitro. I can't imagine a cell in vivo constantly losing big chunks of itself as it moved along, or else it would just keep getting smaller and smaller. That doesn't seem evolutionarily favorable for a cell to do. So I think that losing a chunk is mostly in vitro. Okay, so protrusion, attachment, translocation, and release. That's cell crawling. And I'm not going to play this. I don't think it works anymore. But this is all controlled by chemoattraction, which is what we talked about in chapter 13. The chemical activates the receptor, which activates the G protein, and that activates ARPs. And in this case, it's pulling CAPZ off to open up the plus end so you can polymerize. Oh, and here's another video. This is a pretty good video. I don't know, you can watch that video later. I don't think we need to watch it now. But this is a white blood cell crawling amongst other blood cells looking for a bacterium, and eventually finds it and grabs it. Okay, so that's cell crawling. Any questions about that? Comments?
What were the 4 steps of cell calling?
Protrusion, okay, making your lamellipodium, attachment of that lamellipodium, translocation, which is the stress fibers pulling you forward, and then letting go at the back end, release.
Slish, I have a question.
Yep.
So when the back end is getting released, where is it going? Or is it just kind of like getting pushed back to the front to keep moving?
Yeah, it's the back of the cell is being pulled forward by the front of the cell going forward, right? It's just like the cytoskeleton inside is pulling the whole thing forward. That makes sense?
Yeah, thank you.
Okay. Okay, amoeboid movement. This is kind of simpler. It's a lot less complicated than cell crawling. I think it's a slower movement though. It involves these pseudopodia or false feet that get pushed out. I think they call them pseudopodia because it isn't like a foot that's always there. It's like formed and then goes away. It's a false foot. So this is a pretty simple process, and it all has to do with polymerization and depolymerization. And they've given specific names to the microfilaments depending on their condition. So you've got the sol phase and the gel phase. Gel is like solid. Sol is in solution. That's what used to confuse me. Sol, but it's not solid. No, sol doesn't stand for solid. It stands for solution. So you've got monomers of actin that are in solution, and they're inside the cell. So they're called endoplasm. Okay, endo, inside. So you've got your endoplasm inside the cell, and then along the outside, we've got cell cortex. And the cortex is gel, right? It's solid, and that's holding the plasma membrane in shape. So you've got the gel phase, which is solid, and then the sol phase, which is liquid. Now, when the amoeba gets, or macrophage gets stimulated to move in this direction, what it does is you get contraction back here of actin and myosin, contracting a bit, forcing those actin monomers through the gel and out to the front. When they get to the front, they push the plasma membrane out, and it's really just kind of loose jelly-like stuff pushing that arm out. And then it polymerizes. Now it's solid again. And back behind that new polymerization, you've still got gel. That gets depolymerized. So that becomes sol actin. And then it just repeats. The sol gets pushed through the gel, polymerizes, the gel behind depolymerizes, and you can keep moving in that direction. And that process of converting back and forth is called gel solation, which is great. I love it when a process tells you what it actually does. You don't have to memorize something. Gel solation. And here's an enzyme we've seen before, gelsolin. If you remember gelsolin, it cuts microfilaments in the middle, and that allows them to fall apart really easily. So gelsolin is really important in this process because once that sol actin goes through, the gel is broken up by gelsolin, and then it all falls apart, and then you've gelsolated. Okay.
So gelsolation is the process of the entire amoeboid movement?
Right. It's the process of gel being converted into sol and sol being converted into gel, and that's what causes the amoeboid movement. Here's another figure kind of cut this way through that pseudopodium. In the middle, you've got gel-like actin here. Oh, shit. I didn't mean to do that. You've got gel actin here. You got a little hole in the middle, and your monomers are pushed through. Once they are, this gel behind gets depolymerized, and you get polymerization here. Pretty simple. Okay, any questions, comments, discussion points? What are the ethical considerations of gel solation? Do you feel comfortable with gel solation? Or are you offended by it?
What is it?
What is it? Gel solation? That's the whole process we're talking about. Conversion of gel to sol and sol to gel. And
that can you go into a little more detail? I mean, so is it like, so it turns from a gel to a solid? Are you saying?
No. No, so the gel is the solid, sol is in solution. Okay, so sol is the monomers because they're in solution. So
Oh,
back here is your endoplasm, and the endoplasm is sol phase, solution, gets forced through because it's liquid, and then the gel phase is the solid phase. I know it's confusing. I hate the word sol because it always throws me off. At least it did for the first 10 of the 27 years I taught this. When you've
Liz,
taken this
yes,
course 20, when you've taken this course 27 times, you will understand it too.
Liz, could you give an example of when this process would take place?
Oh, let me show you. Let's do this. What? What are you doing to me? The video's unavailable. Damn it. Damn your eyes. Boy. I'm going to share this with you in a second. I'm just looking up. Oh, here's an amoeboid movement. Stop share. New share. So this, I think, is a macrophage. And it's moving. I think this is its target over here. Bacterium. And you can see like the endoplasm pushing through. All right, that's boring. Amoeba. Eating. All right, this is the bad boy. This is the one we wanted to see before. So this is speeded up a bit, but oh my God, can you see the endoplasm flowing in this way? And this one's coming around this way. We're going to circle that little sucker, and you are lunch. So as for an example of why this happens, this is an example here with the amoeba swallowing something up. And now it's going to pump the water out of that vacuole that formed, and it will get smaller and smaller, and then it will fuse it with some endosomes, and it'll become a lysosome. Help me, help me, help me. Ah! It's the screams. That's why I don't do research anymore.
So the thing that got trapped in the middle, that was a saw?
Sorry, I'm sorry, what? Thing that got caught in the middle?
Yeah.
So this is, I don't know what it is, but it's something that the amoeba is eating.
Okay.
I don't know if it's single cellular. I think it's single cellular. Yeah, it must be. Yeah. This looks like a Chlamydomonas over here. Maybe that's what this is too. I don't know. It's tasty. What's that?
The process we're talking about though is occurring in those arms, right?
Right, exactly. Yeah, let's take a look at that. So you can see the endoplasm flowing out, pushing out the arm, and then it's polymerizing to get stiff again. See how it's stiff on this edge and on this edge. And they meet, plasma membrane fuses to itself, and now you've got a vacuole inside the cell.
The sol is the part that you can see flowing really fast, right?
Exactly. And the gel is the stiff part on the outside, so you don't even notice it. Just remember, sol means solution. So it's actin in solution. I have a chat. The title says it's a paramecium cell.
And what is the thing that gets diffused into diffused out of the paramecium?
I'm sorry, the thing that what?
What is the little liquid that gets diffused out and makes the cell like smaller or bigger?
Oh, so you got to figure this thing's in a water solution. So when it swallowed up this paramecium, it took water with it. And now it's just going to pump that water out. And so the vacuole is going to get smaller and smaller and smaller as it pumps it out.
Okay.
Yum. What is death? I don't know. Okay, that's about it for this chapter. Any questions? Or if not, I'll open it up to the floor for review.
I have a question that's related to what you were just teaching and to
Mhm.
some of the exam review. If for some odd reason the amoeba was no longer able to depolymerize, would it just sort of solidify into a block?
Good question. So what if we added latrunculin A? What would happen? Well, latrunculin A, this is the way the questions come out in the book, in the test. So latrunculin A causes depolymerization. So all the gel would turn into sol and it would become a round ball. What if we added, what is it, phalloidin?
Phalloidin, for example?
Yeah, phalloidin stops the depolymerization. So the cell would go, the endoplasm would go through, it would polymerize, and you wouldn't get depolymerization behind. It would just, the whole thing would stiffen up and it would stop moving.
Okay, great. Thank you.
Okay. Okay, so you guys want to do a quiz? I've got tons of quizzes I can throw one out there. All right, let me find a quiz for you. What is the difference between vincristine and vinblastine? They're two different chemicals, but they do the same thing. So, you know, the difference is they're just structurally different, but the function-wise, they do the same thing.
Where did we learn about those? Are those words we've heard before?
Yep, they are, when at the end of lecture 1, I talked about drugs that affect microtubules. So there's colchicine that binds to the monomer and gets added, and then you can't add to it again. Vincristine and vinblastine, I think they sequester monomers, so you can't make more. And then there's Taxol that stabilizes the microtubules.
Is colchicine used to treat gout? That's
Yeah, I
wait,
have no
what is
idea. I have no idea why.
yeah, what
I have no idea why, but it is. I don't know what the mechanism is.
what is gout just basically like an overproduction of cells?
No, gout is when you break down one of the amino acids, I think it's the purines. When you break down purines, you end up with a chemical, oh God, uric acid. You end up with uric acid. And so basically they say it's the disease of kings, because if you eat too much meat, you end up with uric acid building up in your joints. And that's, you know, that's what caused it. Now, I don't know why colchicine helps with that, but it turns out it does, I guess. Okay, so here's a class quiz we took in 2008. Let's see what the heck this is. Ooh, this looks hard. All right, let me take out some of the spaces and then I'll show it to you in a screen share and I'll give you a few minutes to look at it. Well, one question is something we've already talked about, I think, but the other one is pretty new. Oh, wait, no. No, sorry, that's, things change over the years, and that question involved some stuff that we haven't covered yet, so. All right, this is better. Is that it? I know it doesn't make sense to you right now because I'm doing stuff behind the scenes, but
Hey, Swift, on Moodle, it says exam 1 is on the 23rd.
It depends on where you look on Moodle. I had to change it, and I don't see, this always happens to me. I had to change the timing of the exam because the semester got pushed back a week, and I'm not sure if I changed it everywhere it needed to be changed. But bottom line is we're having a test Thursday.
Okay, sounds good.
Because I
This
got nothing
Thursday.
else. Because at this point I got nothing else to talk about. So Samantha, how would you differentiate between them on an exam? You don't have to. You can use either or both. It doesn't matter. They're the same thing, basically. Sorry for the delay in that answer. Okay, so let me give you this quiz. Okay. Collapse the ribbon. Make it a little bigger, perhaps. Okay, can you all see that? Okay, I'm going to go away for 15 minutes and I'll be back. We'll talk about the answers. All right. Okay, I'm going to cut in a little early here because I read over these questions and there's a lot to talk about here. And these are good questions. I'm glad I brought these up because we can discuss how you're supposed to navigate questions like this and how are they answered. So the first one, fungus-derived cytokeratin B binds to the plus ends of microfilaments and blocks polymerization. How would this affect the structure of the cell? Now, why is the easy question. It's because it's going to cause depolymerization of the actin microfilaments. How will that affect the structure of the cell? Well, what do microfilaments do? So that's why you want to answer this. Usually what the question is like two different facts that you learned in class and how do you put them together to make sense out of something. So if they're binding to the plus end, they're going to cause depolymerization of the microfilaments. Probably the most important thing is your cell cortex, right? The cortex is holding the plasma membrane in shape. So if you depolymerize that, the plasma membrane is going to lose integrity. It could rupture, it could open. You know, you could go on and on from there, and I would give you credit for it, basically. This is all hypothesis generation. So as long as it's, you know, hypothesis, what's a hypothesis? It's a,
An educated guess?
educated guess. So as long as it's educated, you know, it's not like crazy out there stuff. As long as it makes sense with what we learned in class, I have to give you full credit. That's the law. That's the law. Okay, number two, you're a pharmacologist. You should be so lucky. You're a pharmacologist studying a new anti-cancer drug. The model you are using is the squid giant axon. By opening the body case, you can directly observe vesicles moving down the axon via motor maps. You note that low concentrations of the drug slow vesicle movement and higher concentrations stop it entirely. What is a possible mechanism of action of this drug? So the note at the end is what I want to talk about. I use drugs a lot and kind of hypothetical drugs, like it's not a drug you learned about. It's just like, what do you think this drug does? That's really what pharmacology is. It's like you find a chemical that does something, and then you try and figure out how exactly it does that thing. So the basic philosophy behind it, or the basic mechanism is a drug is a small molecule, is going to diffuse into that cell, bind to some protein, and mess up the function of that protein. That's all it is. So what this question comes down to is what makes motor maps move down microtubules? Go back to that drawing I talked about with the head comes up, it goes forward, it binds. Head comes up, goes forward, and combines. So what could a drug be doing in that situation? It could be binding to the microtubule and blocking the interaction of the myosin or the kinesin or dynein head with the microtubule. Okay, that's one possibility. So you might want to look at, does it affect both kinesin and dynein, or just one or the other? It could be binding to the kinesin itself or the dynein itself, right? Could be binding to the head where it actually binds to the microtubule. Could be binding to the ATP site, so it can't get ATP anymore. It could be, you know, binding to some of the structural proteins that keep it attached to the vesicle, and it could be disrupting that interaction, causing them to release from the vesicles. So basically, there are dozens of answers to this question. The bottom line is you're thinking, what proteins are important for this process of the vesicle moving down the microtubule? And any one of those proteins could be impacted. And the educated part of the guess is, what are you doing to that protein and how does that cause the results that you're seeing here? So my guess would be low concentrations versus high concentrations. So you are adding this drug, it is binding to the microtubule and blocking the ability of the motor map to grab the microtubule. So at low concentrations, drugs are always bouncing on and off. Every now and then a site opens and the thing can grab and move forward. High concentrations, that microtubule is saturated with drug and there's no way that motor map can grab onto the microtubule. That's what I would guess. But again, there are dozens of answers to this question. You just have to make it rational based on what we learned in class. Number 3, I'm going to skip because I might put that on the exam. I always put that on the exam. I don't know why I gave it to you here. Okay. I think it's one of the questions in the in the questions I gave you in Moodle. So we'll leave that up to you. Okay. Oh, now here's one that's got a gajillion answers to it. This is the thing I like to do because a lot of times people who are just memorizers will read this question and say, I don't know, and just get stumped. You want to be more than a memorizer. You want to be a thinker. So in studying the PDGF created movement of human fibroblast cells, this is what we're talking about today, growth factor causing cells to move. Cell crawling. You note that a mutant cell line has lost the ability to respond to this growth factor. So a mutant cell line, when I say a mutant cell line, what I mean is there's one protein that's mutated and no longer functioning correctly, okay? So keep that in mind because I use that term a lot. So mutated cell line means you've got this cell line that's got one mutation in it, one gene's been knocked out or changed in some way, and now it's changed its ability to respond to the growth factor. The cell has receptors for it, okay? So the PGF is binding to receptors, but don't move when stimulated by it. What changes in the cell would you look for in addition to movement? What 3 proteins may be mutated to result in this loss of movement? Okay, so what would you look for? They're not moving. So do you see lamellipodium? Do you see stress fibers? Do you see filopodia? Are all those structures there available? If you see no lamellipodia, what proteins do you think might be mutated? Could be the G protein, could be WASP, could be ARP2 and 3. There's 3 proteins there. What else? If you don't see stress fibers, maybe the myosin is mutated. Maybe the G protein involved in stress fibers is mutated. If they're not moving, it could have something to do with the focal adhesion. Could be any one of those proteins involved in the focal adhesion, the integrin, the talin, the alpha actinin, all of those things. Any one of them could be mutated out. What else? If it's not moving, well, that's a lot of it, but hopefully you get the gist, which is basically, what are all the things involved in cell crawling? If one of those is missing, if one of those proteins involved in it is missing, it's not going to crawl. It needs everything there. So again, all these questions, with all of these, maybe not so much number 3, but with most of these questions, there's a lot of different answers you could give to it. It just has to make sense. Okay. Any questions? Oh my God, I still have 29 people. I thought everybody was going to leave. 12:09, almost filled the whole class period this time. So any questions or comments? Robin Russell, any questions?
No, I don't have any questions.
I'm sorry, I have to tease you. I only tease the people I like, so don't be upset. Okay.
Somebody asked a question in the chat about, do you know what the test layout is going to be yet?
Yep. I think there's a sample test up there from last year or from 2019, which is a paper test I gave in class. The test you'll do on Moodle will be similar in that the first half of it, 50%, will be just this memorization stuff. The game should help you with that. Don't forget, I gave you that game last night. Don't forget to do that before the exam and hand me in your score, because those bonus points go right on your exam score. But anyway, the first half will be like that, just memorization, what drug binds to the plus end of the microtubule and blocks polymerization. Boom, and then the second half will be questions like we did today, just more complicated, more like synthesis of the knowledge rather than just regurgitating it. And there are examples of that second half in that test I gave you. There are practice questions. I think they're called quiz questions that you can do. So those will help you understand how to answer those questions. Okay, anything else? Trevor DiCarlo's in this class.
Hell yeah.
That guy's trouble.
Yo, actually, I was meaning to ask you, can we make are you open for office hours tomorrow?
Let's see, I've got a class at 2 o'clock. No, 1 o'clock. Other than that, I can Zoom with you if you want.
Hell yeah, because I need a half academic advising / cell biology refresh.
Okay, I'll send you an email. We'll set that up. Are we signing into Zoom while we take the test? No, it's just on Moodle. It'll be the same time as our class time, but you'll go there. And you can email me. I'll be available if you have got questions during the exam. Brian Pinello, guess what, buddy? I'm on Ethernet today. No more cutting in and out. You're the man.
What?
You need Verizon.
Verizon?
Yeah.
I'm on Burlington Telecom. I think I just recently went from something like 5 megabytes per second to 150 megabytes per second, so I should be fine. I've got like a fire hose of bits coming into my computer every minute.
Well, that's awesome. Yeah, no, because Plattsburgh, it's Spectrum dominated here.
Yeah.
But Verizon at the beginning of last semester, they just started serving the area. So I was like, oh, yeah, we're going to do this. Came to my apartment, drilled a hole in the wall to put the fiber optic thing in there. We got 1,000 megabits per second. It's crazy.
More is better.
Hell yeah.
Okay, no questions, comments. We're gonna call it a day.
Bye.
Peace.
Don't forget to do the quiz or the game and send it to me. Talk to you later.
Yep, have a good one.